Um, so first of all, let's get electron transfers down. Take a breath. Exhale, okay? You just made an electron transfer reaction, okay? Didn't feel too bad, did it? You just breathed in oxygen from the room, and you breathed out water and carbon dioxide. So you converted the oxygen to water. That's an electron transfer reaction. So you and I are hydrogen cells that are converting bond energy from the food we ate into energy that allows us to live. And every organism does that. Every organism does that. So one of the very first things that Vernadsky, in his prescient way, realized very early in the beginning of the 20th century was that every organism exchanges a gas with the environment. And we use that information to inform us, in the extreme sense, of whether or not there will be life on an exoplanet. Okay? Now, I obviously made the term light to life as a little bit of a tongue-in-cheek, but it's reality. So you and I are stored photon energy bonds. We didn't make ourselves. Plants made us. And let's go through this in a way. I'm just going to start out with the acknowledgments. There are many people that have worked with me over the last several years. Jana is in the audience here, Jana Bromberg. She's a bioinformatics person. Dave Case, some of you may know. He used to be at the Scripps Institute. He's a biophysical chemist. Also, Ben Jelen is here. He's a graduate student working with me, and I'm going to talk about some of his work. But a lot of the work that was done here that I'm going to present today was done by a former graduate student, John Kim, a little bit by Stefan Sen. John is now in Korea, back in Korea, and Arik Harel, who is back in a position in Israel. And my old buddy, Vic Nanda here, who is a structural biologist. Now, let's begin at the beginning. So this is a very classical slide. It's from Goldschmidt. Victor Goldschmidt spent his entire life basically trying to reconstruct the origin of elements in the universe. And in our neck of this universe, by the way, Martin Rees is in the audience. He's the astronomer, you're still the astronomer royal. He's the astronomer royal in England. So all the astronomers claim that everything higher than lithium, they call a metal. So I don't know why, but they talk about the metallicity of planets, and those are metals. Anyway, carbon and oxygen can be slightly different in their relative population densities, but you see there's hydrogen. Let me get the low-tech, no battery-powered required elements. There's hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur. We call those the big six. So those 6 elements are responsible for all polymers of life, the amino acids, the nucleic acids, the lipids, and the carbohydrates. 6 elements, of which 1 was created in the original Big Bang, that's the hydrogen. All the others are created in supernovae, not our star, but a very hot star that exploded before our solar system was formed. And the dust particles from that explosion were swept up into our solar system and other solar systems nearby. But the point is this. We walk along this, and you see the odd, even numbers here, the zigzag pattern. So you have even numbers of protons, odd number of protons, even numbers of protons, so on. These are fusion, fusion, fusion reactions. And we get over to here, chromium. And chromium with hydrogen-4, through so-called alpha assimilation pathway with helium, can form another very, very important element, iron. So iron becomes the most important transition metal on Earth. Now, this is what I just said, the big 6. Now, let me just explain to you what I'm going to talk about for the most of this talk today. Hydrogen, carbon, nitrogen, oxygen, and sulfur undergo electron transfer reactions in biology. Phosphorus does not today. Phosphorus is stuck in what we call acid-base chemistry. It's just phosphate. You know it when, in the old days, the bad old days, you had it in your detergents, your laundry detergents. It's a very, very nice nutrient to grow algae in. Nitrogen, the most abundant element in the atmosphere, is in the form of an inert gas. N2. It's useless in biology. In order for it to be used, you have to add hydrogen to it to make ammonium. Carbon is primarily in the form of an oxidized species, carbon dioxide. In order to make that usable, you have to add hydrogen atoms, which have an electron and a proton. So let me be very clear so we're not confusing things. This is so-called oxidation reduction chemistry. It's the movement of an electron with or without a proton. Okay? And sulfur similarly has various so-called redox states, where sulfate is the most abundant in the ocean today, but hydrogen sulfide is that smelling of rotten eggs type of gas that you see when you go down the beach. Those are made by organisms that are converting organic matter into something for themselves. They're respiring it, and they're looking for a sink for their electrons, so they're dumping their electrons onto sulfate to produce hydrogen sulfide. So these are processes of biological systems. So let's just talk about this for very, very quickly. Life is electric. This is not a metaphor. Okay? It's not a metaphor. It's an absolutely true phenomenon. Life is electric. You know this, by the way, for yourself. Anybody who's ever had an EKG knows you're electric. But you can do in any microbe, if there's no electrical field across a membrane, the cell is dead. The cell has to have a membrane potential. So all organisms derive energy for growth and maintenance by moving electrons from a substrate to a product. And it follows there that the substrates and products must be cycled. So if there's one organism left on Mars, one microbe left on Mars, it's doomed. Biological processes, therefore, are paired. So we can talk about the trivial ones of photosynthesis and respiration, which I'll get to in a minute. They're not so trivial, but they're relatively trivial compared to some of the more esoteric ones. So biology has created on this planet what I call an electron marketplace. They're buying and selling electrons. Maintaining life, then, requires a recycling of electrons, and what you see now is not what we started with. So the Archean Ocean, which is a period of time from roughly 4 billion years ago, the Earth is 3.56 or so billion years old, 4.56 billion years old, up until about 4 billion years ago was very heavy bombardments. We had many, many, many meteorites hitting this planet. It settled down around 4 billion years ago. And up until about 2.5 billion years ago, we call that period the Archean, the old period. There was no oxygen, or very, very, very little oxygen then. So in the Archean oceans, the sources of electrons were hydrogen gas, iron, which was in the form of ferrous iron. This is not fully oxidized. This is not rust. H2S, which I've described to you, that rotten egg-smelling gas, and carbohydrates, sugar. Today, and by the way, so this is for scale me. I'm about 1.6 or I don't know, something meters. Not even, my wife would say. And here you can see these are from Ishua Formation. These are 3.8 billion year old rocks. I'm just pointing this out to you. Every rock in the Ishua Formation of Greenland is about 3.8 billion years old. If you go through these seams, deep in, you see oxidized iron. There's no oxygen yet, so just keep that in mind. Today, the source of electrons is water. So we know that the Earth is covered by a thin film of water. For those of you who are students in the audience, if I had a meter and I wanted to ask how thick is the film of water on the surface of the planet, it'd be about the thickness of one page of paper in that meter. It's a trivial thin film, but it gives life to this planet. By the way, this is an actual photograph. Now, before I get into the deeper stuff here, let me explain to you the oxygen in this room that you're breathing came from splitting water by a photosynthetic organism. The driving force for that was light. solar energy. Now, as far as I know, there are no trees in the room right now, and as far as I know, most of us are not photosynthetic. But you're still breathing oxygen in the room now. And if you went to Antarctica in the winter, you could still breathe oxygen, even though the sun isn't going to shine for 24 hours a day for a few months. So how did the oxygen get there? It got there because we have an atmosphere, right? Obviously. So that's one of the two main wires on the planet that connects biology. The second is the ocean. The ocean and the atmosphere are two fluids that act like wires that allow metabolism from distant parts of the world to be connected to another part of the world. So we could make oxygen in Japan and 1,000 years later be breathing it in Rome. You can exhale the carbon dioxide, and that carbon dioxide will be consumed from New York City somewhere in France. You can't trace it. But the atmosphere is a wire. Now, I'm just going to show you a little bit of what I mean by nanomachines. So this is the most complex energy transduction system on the planet. It's the system that makes oxygen. It's got little nanomachines. I just want to explain to you one little part of this nanomachine. This little nanomachine has 4 manganese atoms sitting in a little cluster down here, and that's where the work is done. The electrons are removed by oxidizing a chlorophyll. The chlorophyll says, okay, I need an electron now. I just lost my electron. I just gave it to somebody else. Where is my electron going to come from? Oh, there's a manganese atom there. Okay, I'm going to rip out, I'm simplifying it. I'm ripping out an electron from the manganese atom, and the manganese atom says, wow, wait a minute, I didn't like that. That wasn't fun. I need an electron. And it turns around, there's a water molecule there. It says, oh, pop. Not such an easy trick. Now, it does this 4 times sequentially, and then it produces this waste product, this gas, O2, which just diffuses out of the cell. The electrons merrily go on their way through this thing called cytochrome B6F to another reaction, yada, 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 and then we make carbon dioxide into a sugar. But let me just explain very quickly so you understand what these machines are like, because I do these experiments. In this part of the machinery, we're ripping an electron from a chlorophyll molecule, putting it onto another molecule within that box of proteins. So I have a positive charge here, a negative charge here, separated by about 90 angstroms. That box goes like that, like that, like that, like that. Every single photon that comes in, like that, like that, like that, like that, and I can synchronize them with a laser pulse. And I can measure how hard they clap. It's called the photoacoustic effect. And the clapping sound in dB is directly proportional to the amount of energy stored. It's amazing. It's a very beautiful biophysical experiment. It's the only machinery in nature that I know that does that. This one does the same. Now, this is another machine which is cool, but I'm not going to go into it today. This machinery allows the protons to flow through, and then this rotor turns, and this goes back and forth, and that's how we make ATP. So these nanomachines evolved many, 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 many billions of years ago, way before, in the Archean. Okay, now, over time, life became more and more complicated. The first organism that ever made oxygen was appropriated by many, many other cells by what's called symbiotic associations, endosymbiotic associations. And we come down to the world we see today with all these algae like diatoms and boring things like plants. Frankly, as one eukaryote speaking to another, and I'm going to be very frank with you, eukaryotes didn't invent a damn thing. All we are different body plans of prokaryotic metabolism. Now, a few years ago, I was working on how to make what is essentially a KEGG diagram. For those of you not familiar with this, this is the Kyoto Encyclopedia of Gene and Genomes, which is an inventory, if you will, of the genes that are responsible for metabolism. And I wanted a wiring diagram of Earth. So together with my friend Tom Fenchel, who's now retired, and Ed DeLong, who used to be at MIT and has the good sense to go to Hawaii, we developed a wiring diagram. So this is the wiring diagram of all the major electron transfer reactions on Earth. Most of them are anaerobic. These are all driven by photon energy. These are all respiratory pathways that are consuming electrons. They're oxidizing reactions, like you and I. And then I challenged Ben Jelen, who's here, to essentially do the hard part, which is to annotate that. So he divided this up into various metabolic domains, and these are the metabolic domains. But at the end of the day, and I'm not going to show you the results of this, at the end of the day, it turns out there are only about 400 genes that move all the electrons across the planet. So you have 2 wires, 400 transistors, 1 power supply. Shouldn't be too hard to reverse engineer that. Shouldn't be. I mean, we're pretty smart animals. So I took this as a literal problem. I took this as a literal problem of a circuit board. I considered Earth to be a circuit board. I'm not really interested in the organisms. Organisms are transient features on a planet. They're around for 3 million years, 4 million, 5 million years. It's the genes. As long as you don't lose the genes, it doesn't matter what the body plan is. Okay? So I'm not going to follow organisms here. I want to know how the electronic circuits are designed with the wires, the power supply, to create that network. So one of the first things we did very early on, and Jana was very instrumental with this, with Arik Harel, was to do an inventory of what are the major metals that are responsible for the electron transfer reactions in the enzymes that are doing the electron transfer reactions. And no big surprise, approximately 65 to 70% contain iron. And then we can see a large fraction contain copper, and then manganese, nickel, and molybdenum. Doesn't matter so much. But we focused on this basic distribution. And we were inspired, at least I was, by a person, unfortunately, I never met. She died long before I was really cognizant of what was going on in the field of structural biology, and this Margaret Dayhoff. I think Dayhoff worked at Columbia for a while. She was one of the very first computer scientists, and that's why we have single-letter amino acid codes, because she was punching cards. And it was easier to punch a card with a single letter than to make 3 letters. Okay? But she wrote a paper in Science when she was looking at, by inspection, a molecule which I'll show you in a minute called ferredoxin, which moves electrons. And she realized it was a dimer. The original molecule must have been 28 amino acids. The molecule she's looking at is 56 amino acids, and it was a gene duplication event. But what she said is, why didn't this change? Why is it ferredoxin, a ferredoxin, a ferredoxin everywhere? Why isn't it changed? I can go to a lowliest bacteria up to an oak tree, to a human being. Ferredoxins are ferredoxins. And so she wrote this with Richard Eck. The processes of natural selection severely inhibit any change in a well-adapted system on which several other essential components depend. That means once you made the machine, even if it's imperfect, everybody depends on it, don't monkey with it. You can't monkey with it. You may have a better idea for making another keyboard, but the typewriter is the typewriter now. Now, this is ferredoxin. It's got a core of an iron-sulfur cluster, 4 irons, 4 sulfurs. And every ferredoxin is bound by 4 what's called cysteine residues. These are iron-sulfur clusters, sulfide clusters themselves in amino acids. So there's cysteine, XX cysteine, XX cysteine, and then another cysteine. Now, in every single one of these, the iron-sulfur cluster has no chirality. There's no left-handedness or right-handed to it. In every single iron-sulfur cluster, you could have grabbed it this way or this way. Every single one in nature is this way. Why? Well, it's a very nice paper that John Kim wrote a couple of years ago. When you're given L amino acids, there's one more hydrogen bond that's allowed for the stability of that prosthetic group. If you had been given D amino acids, it would have been left. So it's just a funny thing that we have L amino acids in nature, and the chirality is very widely discussed, but they're all right-handed folds because of that, because of the increased stability of of the hydrogen bond. And then you can address these issues a little further on. You can make many more iron-sulfur clusters and get more fancy. And in this particular case, there's one molybdenum atom that was added here in this enzyme. It's not necessary. You don't have to have it, but it is there in most of them. And this enzyme is the only enzyme in nature, the only one, that can take N2 gas and make it into ammonia. And we still don't know how it works. So Tom Schrock has worked on that for a long time. How do you get so many electrons moving is very, very, very hard to understand. If we can understand how this reaction works, we can change a lot of energy supply to the human population, because about 23% of all agricultural energy in the planet is used to fix nitrogen by a very inefficient process called the Haber-Bosch reaction. And then we have various other types of proteins, as you see, with their various prosthetic groups. Okay, I don't want to bore you with this. They're nice pictures, but the point is this. How do we figure out how nature designed this stuff? That's the origin problem, right? How do we get to figure it out? So Ulrich Arell, a postdoc with Jana, went back into the protein database and other databases, not just the PDB at Rutgers. SwissProt, there are other databases he used. And that's an aside. The point is that of all the 400 or so proteins that are responsible for transferring electrons in life, there are really only 35 motifs. We just collapsed 400 down to 35 transistors in the box. Now, that's pretty cool. I like simplifying things. I grew up in physics, kind of. I don't like more stuff. I like less. So here's what John Kim. This is a really, really cool idea that John Kim did. And this is one of those kids when you have a graduate student who's fearless but also really smart. He can be dangerous. So John Kim has his degrees in chemistry and maths, and he's really good in statistical mechanics. And so I said, John, how do we arrange this stuff so we're not going through top match and 3D stuff all the time and trying to figure out how these proteins look at it or relate to each other? So he did this. He said, look, I'm going to go into the center of each of these metal clusters in each of these proteins, in all of them. And I'm going to walk out, and he did the experiments to see what the sensitivity was. I'm going to walk out 15 angstroms from the center in all directions, make a sphere, 16 angstroms. And then I'm going to say, how much of this is a sheet? That's one of the major types of secondary structures, a loop, or a helix. So those are three big secondary structures in proteins. What percentage of them? And then I'll just array them in this two-dimensional space. So every protein has a point of up to 0% loop, 100% loop, 100% sheet, 0% sheet, 100% helix, 0% helix. Now, from each of these points of every protein, I can make a distance map, a Euclidean distance. And from a Euclidean distance, I can tell how far away they are from me, objectively. I'm going to make one Bayesian prior here. I'm going to make the prior and we can talk about this if you like, that inherently disordered proteins are the most evolvable because they have the fewest hydrogen bonds. So the loop clades, the more loop that you have, that's the most evolvable that you have. And so we can arrange this energy space with the amount of hydrogen bond energy in each of these. And we see that there are some places that are highly populated and others that are not. But at the end of the day, here's what you get. This is the reconstruction of the phylogeny of the electron transfer reactions that are extant based on topology, not on sequence. So the oldest type of folds is the iron hydrogenase or the ferredoxin fold, very similar types of folds. That's that iron-sulfur cluster that contains cysteine, XX, cysteine, XX, cysteine, okay? That's where we begin at least to think about. This is only around 8 or 9 amino acids, and you can already have something catalytic, potentially. And then we walk out, and this is the loop clade. You can see these are very old proteins up here. This is the sheet clade, and then we wind up here with the helix clade, and this is the cytochrome C oxidase. That's the prototype of the enzyme in your mitochondria that is allowing you to breathe oxygen. One of the very last folds to evolve. Now, we can array that phylogenetic tree in terms of the distribution of metals that are used, and you can see various patterns. And I'm not going to go into this. We published this a few years ago. But the point is this. It's not easy to say, and I think it would be incorrect to say, that just because we had a reducing ocean, we didn't have molybdenum in proteins or copper. Almost for certain, you had those metals, even though the ocean was reducing, and they're not supposed to be, those metals are not supposed to be abundant. And I'll explain to you how we get them there. Now, let's go to the bottom-up story. So here's the, I haven't talked to you about light yet. All this is nice, but that's the top-down approach of looking at how proteins evolved. Let's take a look at another approach, totally different, bottom-up. So in the present atmosphere today, the sun is shining, and the solar radiation coming into the top of the atmosphere is given by this black line. And the top of the ocean is this red line. Now, there are all kinds of absorption bands that are missing here, but look at this. This is ultraviolet radiation. It's coming in at the top of the atmosphere, but it's not hitting the surface of the planet anymore. Why not? Because we have a gas that is protecting or shielding this planet from ultraviolet radiation. That gas is ozone. Ozone can only be made, as far as we know, on any planetary surface by the production of oxygen, O2. Now, ozone is not a meter of oxygen, but it has to be, oxygen has to be present for ozone to be produced. Now, for reasons that I'm not going to bother you with, which are somewhat arcane, we know that the ozone came into play somewhere around 3.4 billion years ago. It's called the Great Oxidation Event. And we know that because of the change in the sulfur isotope fractionations. If anybody wants to talk about this, is a very arcane subject that Roger knows very, very intimately. I know somewhat intimately, and it is a very interesting story. But anyway, to make a long story short, in the Archean Ocean, there was much more UV light hitting the planet. And why is that important? All right. An experiment. I'm an experimental guy. I don't like, theory is great, but you know, I'm a show me guy, show me state, whatever it is. This material in this quartz cuvette is siderite. Siderite is a very simple molecule. It's iron carbonate. You're all familiar with calcium carbonate. That's calcium carbonate. In the Archean Ocean, iron was very abundant, and it would have precipitated with a huge amount of bicarbonate in the ocean to form this mineral, siderite. Siderite, by the way, is destroyed by, not destroyed, but it is an aqueous phase. It's very unstable in molecular oxygen. So this is precipitated under absolutely anoxic conditions. Now, we calculated from what's called density functional theory that there's an anti-bonding orbital. Though it doesn't matter what that is. All it means is I can pop an electron out of that iron 2, and I can make it into rust at 270 nanometers of light, UV light. And so this is an example. We just shine UV light on this, and we generate a population of oxidized iron. No organism necessary. No oxygen required. All we did was drive an electron out of a metal with a photon. All right. When you do that, you generate a new species, which is magnetic. I'm not going to bother you with all those details, but siderite is not magnetic. And it's a 2-photon reaction. We generate 2 photons. Now, if you're a chemist, if you're oxidizing something, something has to get reduced. Where does the electron go? It goes to reduce protons to hydrogen gas. We just generated hydrogen gas by oxidizing siderite. Now, there are very, very few geologists that play with light energy as a driving force of moving electrons around. It went out of fashion. When it was in fashion, there were 2 people that did it. Let me explain that we know there are a lot of, from density functional theory, a lot of very common minerals that can be oxidized by UV photons very well. So I can generate soluble molybdenum or copper without any biology. I don't have to have oxygen. I just want that to be somewhat clear. The second point is this. This reaction is not catalytic. It's consuming siderite, generating iron hydroxide, and generating hydrogen gas, which is fine, but all the reactions that are of this sort are not catalytic. They're sacrificial. And why is that important? Because in our quest to understand origins of life through metabolism, and metabolism must be a process in life, we have to make something that regenerates itself again and again and again and again and repeats the reaction again and again and again and again. So inspired by the siderite photooxidation, I'm now trying to make very small maquettes, we call them. It's a term I don't like, but Les Dutton uses it, so okay. It's a term which means it's an artificial protein in which I can insert a metal or maybe a complex like an iron-sulfur cluster in a very simple protein, maybe 8, 9, 10 amino acids, and go from a sacrificial reaction to a continuous reaction of the earliest catalysts. And what do I want to do here? I don't want to simulate a hydrothermal vent hydrogen production system. Fine, that's fine. At some point, if that's where origin of life began, fine. At some point, you have to crawl out of that hole and use photon energy. And we certainly didn't do it with that very complex machine that I showed you earlier, which is clapping its hands all the time. We did it with something really, really, really simple and probably wasn't very good, but it probably worked. So how much hydrogen can we get? Quite a lot, actually. If you do a very simple calculation, it's about equal to the total hydrogen flux from this calculated serpentinization reactions that occurred during Archean times. Non-trivial. The reason that I appeal to you for this kind of reaction is that hydrothermal vents are transient features. A few tens of thousands of years, then they go away. The sun is not going to stop shining. The sun is continuously supplying photon energy to the surface of the planet all the time. It's reliable, which is why it's the main source of energy on the planet today. So I'm going to conclude. I'm a little bit early, but I think that's probably good for you. In the first 2.5 billion years of Earth's history, I call it the research and development phase. Okay? This is when Silicon Valley sprung up in the Archean world for microbes. There were a bunch of nanomachines that were set up, and they formed a core set. The survivors come to us today. We see them in the ribosome, we see them in the ATPase, we see them all over the place in biology. Those core nanomachines are in every organism. They were all developed in prokaryotes. So if you're studying eukaryotes, you're probably looking in the wrong place. And some of you probably went to medical school and realized that was a mistake. So now you can go back and you can... My brother certainly understands that very well. My brother was a pediatric heart surgeon, and he wishes he were doing oceanography. Okay. All right, so there are only about 400 core genes. And what I want to say to you is a fun thing. These metabolic sequences are not just coupled in a test tube. They're coupled literally on a planetary scale in ways that we really don't understand. All we see are the manifestations of this in terms of the gas composition of the planet, and to some extent, the redox poise of the planet. So let me explain the redox poise of the planet for a second. If you go out, you can't do this easily in New York except when you're in Central Park. But if you go out to Central Park and you are looking at the ground after it's soaked with rain, and if you were to take a probe and stick it into the ground, it would be reduced. The material, there would be very little oxygen in the soil within a few millimeters. You're standing, it's oxidized. The entire surface of the planet acts like one gigantic membrane. There's a redox potential across the entire planetary surface. Now, the astronomers know this for many, many years. I'm not telling them anything new. But that redox poise was set up to first order by biology. To first order. Now, I'll have arguments with many people about whether it was first order, because a lot of geologists would like to have hydrogen escape as the first order process. That happened, but it doesn't lead to that oxidation reduction kinetic or limitation at the surface. Finally, that little machine that produces the oxygen, it has a fluorescence signal in the red. What does fluorescence mean? It means it glows. When you shine a blue light on it, it looks red. Now, believe it or not, we have 2 satellites in space. They're MODIS AM and PM, and their equator crossing times, local time, is 11:30 going to the south and 2:30 going to the north. And they scan the entire globe about once every 3 days. We can measure the fluorescence from the sea surface. Amazingly. And we can actually measure it very precisely. It's the only physiological signal of life on this planet. that you can measure from space, the only one that I know of. Everything is color except for this. You would not get fluorescence if there was not a photochemical reaction. So I'm just leaving you that light is provided for us, life, but it also provides a signal for life, which I think is very fascinating. So with that, I'll stop and entertain any questions you may have. Thank you very, very much.